Next. Welcome to our monthly meeting. Next slide. Okay, so this is the WastewaterSCAN partner call. This is a national research project that's based at Stanford, and it's in partnership with Emory University, and we work with Verily as our contracted partner for running our analyses for us. Also, all of you, without you, this project would not be possible. Next slide. A review of our program is coming in the next slide. So we work in 40 states with wastewater that represents over 38 million people in the US. These are the locations of all of our sites in purple, and the dark gray states are states that have at least one wastewater treatment plant enrolled in the program. We are currently monitoring for a large suite of pathogens, nucleic acids using digital droplet PCR. They are shown here. You will notice two new targets, the BLA-NDM gene, which is an antibiotic resistance gene, and also Mycobacterium tuberculosis TB, now listed under the emerging and outbreak pathogens of concern. And we'll talk more about those in a bit. Next slide. All of our protocols and papers are publicly available, and you will receive these QR codes in the follow-up email after this meeting. And also our website has links to all of our papers. And if you need help finding one, you can always reach out to us. All of our methods are peer-reviewed. So here are some recently published papers in preprint. So the measles paper was finally published, and you can access the peer-reviewed paper at this link. And for preprints, we now have a paper on the prospective metagenomic sequencing of wastewater from the phase A of our project, which Marlene will also cover later in this presentation. We also have a preprint on wastewater detections of pertussis and Mycobacterium tuberculosis. That paper is in the final stages of the review process at a peer-reviewed journal. And we also have a paper where we looked at avian influenza strains in wastewater at a municipal wastewater treatment plant. And you can access all of our papers through this link here on our website. Next slide. We also have a monthly newsletter that we send out via ghost.io, which is a blog site. And you can always go to the blog site and read the monthly newsletters to get an update on where things are at and what we're measuring and other new developments in the program. Next slide. And we have fact sheets and guidance documents to help you interpret what we're measuring in wastewater. These are available on the website, and you can also contact us. A lot of people maybe have never had a detection of some of these more emerging targets and then need some help interpreting them. And we always send people first to our fact sheets, where we've tried to compile a lot of information that we have on the detections. We recently added fact sheets about detection of the antibiotic resistance gene BLA-NDM, and also a frequently asked questions on the TB in wastewater. So you can access those. Next slide. We're going to talk a little bit about the scaling of these assays. So the data went live to the public on July 7th, which was 3 days ago, through our data dashboard following our pipeline or our template for how we scale things. So you all had access to the data for a couple weeks or more before we released the data publicly on the website. So the BLA-NDM gene is a gene that if bacteria have it, it makes them resistant to a broad suite of beta-lactam antibiotics by producing NDM, an enzyme that inactivates the antibiotics. And it is extremely versatile in inactivating a broad class of antibiotics and is highly genetic genetically mobile. So it is an antibiotic resistance gene that is of concern globally. Next slide. And then that's why we chose that antibiotic resistance gene of all the ones that we had been looking at before. And then we decided to monitor TB in wastewater based on feedback from all of you and also other things, which is that TB has been increasing globally in recent years. And it's also a very important pathogen globally. And they can also, TB symptoms can be challenging to identify in the early stages of an infection. And TB incidence is very low in the US, but there have been cases and outbreaks that have been increasing in the past several years. So given your interest and our interest, we thought it would be interesting to monitor for TB. Next slide. The assay for the antibiotic resistance gene is shown here. It was validated previously, and we used it with wastewater scan samples in a project which we've talked about before, and that has been published in Nature Communications, the Kim et al. Paper, Susie Kim, who used to be a PhD student at Stanford. And the assay will detect a whole suite of variants of the BLA and DM gene 1 through 15, 17 through 31, and 33 through 47. I think the variant 5 is one that people are particularly concerned about right now. Next slide. And so here are some of the results. So this is a screenshot from the dashboard, and you can find the antibiotic resistance gene on the dashboard now. And these show data for 2 sites, and I can't tell which ones, but it's not important really. It show actually more than 2 sites, but it shows the concentration of the antibiotic resistance gene normalized by PMMOV on the y-axis versus time. And so you can see for a couple plants that the levels are very low. They're close to 0. And I think, well, it doesn't matter where it is actually, just that there's variability across the sites. And it's in the other pathogen grouping. We are not calculating a category right now because I think we're learning from this experience how to interpret these values and when the levels will be of interest and of note to the community, to you all. And then I just wanted to make a note that we chose to normalize by PMMOV here. Normally, you would see antibiotic resistance genes normalized by the 16S rRNA gene as a proportion of the bacteria potentially that have this gene. We don't have measurements of 16S rRNA, but some analysis we did with data from across all the wastewater scan treatment plants sort of at one moment in time where we measured 16S rRNA, it was positively and significantly correlated with PMMOV. So we feel that this is a good option for normalizing the NDM gene concentrations. Next slide. This is the TB assay. It was previously published, and we have a preprint sort of describing why we scaled the assay or providing an evidence base for scaling it, which is basically that we were able to detect TB in wastewater in a location in the United States that was experiencing an active TB outbreak. And the results suggested that concentrations in wastewater were related to the outbreak. And so with that evidence base, we felt there was evidence to support scaling the assay across the US. Next slide. Okay, so here is the default view for TB. We have not detected TB yet, which I think is good because there's not, to our knowledge, a lot of TB. There's not a TB outbreak that we're aware of, but that doesn't mean that there's no TB infections. But I think if we were getting lots of detections, we would be wondering and concerned about using this assay. So I think this is consistent with what we would expect, no detections. The default view is the heat map, so just showing presence, absence. These are results for Alabama, just for illustration. They're found in the other pathogen grouping. And we're also not, we don't have a category yet for TB. And I think we'll see how it goes and if we do eventually detect it. Personally, I guess I'm a little surprised we haven't had one detection yet. But again, I think probably you all are probably think like that's probably good because you don't know of a lot of TB circulating right now, and the US does not have a lot. So we'll see what happens. I'm curious myself. Next slide. Okay, so for reference documents, we have the two frequently asked questions, the two facts for these, which sort of explain answers to questions that we normally get, like, why are you measuring this? What does it mean if I have a positive detection? And the evidence base for providing the measurements to you for surveillance purposes. Next slide. Okay. Is this where I'm changing to you, Marlene?
Yeah, I'll take
Yes.
over.
Okay. Okay.
Okay, so a couple other ongoing research projects to mention. We have some ongoing work on Oropouche virus that's happening. And then our viral metagenomic method development, which I'm going to talk more about right now. Phase A sites were selected and notified in October, and we completed that pilot at the beginning of this year. I'm going to talk about the results now. And Phase B sites, where we expanded our scope a little bit, were selected and notified in April, and we are currently in the middle of Phase B and continuing to do metagenomic sequencing with the 30 sites that are involved in that phase of our method development and expansion. So now I'm going to talk about this phase A pilot study. This is an overwhelming slide. Don't become overwhelmed. I'm going to walk through it, but I want to describe the methods that we used to do this study, and this is the figure that you'll find in the preprint if you go and read it. So I will walk you through it here, but essentially we worked with a subset of WastewaterSCAN samples that we were already receiving for this pilot. There are 15 sites that were, again, already in WastewaterSCAN across 12 states, and over a period of 12 weeks, we took 3 samples a week from each of those sites. And in addition to doing our normal standard solids digital PCR process like we do with every sample, we also did hybrid capture metagenomics on those samples. So again, 15 sites, and that covers about 11.4 million people who are kind of represented in this subset of samples. For the metagenomics, which is the bottom section of this panel, we used the liquid fraction of the samples. So if a site was sending us a sample from influent, we used the liquid. If they sent us a sample of sludge from the primary clarifier, we spun that down in the centrifuge and used the liquid. So rather than using the solids like we do for PCR, we were using the liquid part of the sample for metagenomics. We then did concentration and nucleic acid extraction. And then for our metagenomics work, we're using a process called hybrid capture. Viruses, viral reads will typically make up less than 0.1%, often less than 0.01% of reads in wastewater if you just sequence everything. So enriching the samples using a hybrid capture approach can help us make sure that we are going to find viruses and we're going to find these rarer pathogens that we're interested in. Hybrid capture uses probes that are designed to capture a really wide range of target genomes. We're using the TWIST viral metagenomics panel, and that has more than 3,000 viruses that are included in it. It also has a lot of divergence in those probes. So it captures a really wide range of viruses, even if some kind of small changes have happened across the genome. So we're using that probe-based hybridization, hybrid capture process to enrich the sample for viruses. And then we're doing Illumina short read sequencing to generate sequences from everything that's in the sample, and then doing bioinformatics to remove any off-target reads and do viral identification and quantification. The main tool that we're using for that is called SVR2. So that gives us viral taxonomy and relative abundance. So that is the short overview of how all of these methods work. Amanda has already linked to the preprint there, and we can follow up with it again. And so the methods are spelled out in great detail in that preprint and protocols that we have posted. So, a little bit about our initial results from the study. So, overall, we analyzed 443 samples, generated 1.2 billion viral reads as part of this phase A effort, and detected over 1,300 unique viruses across all of the samples that we processed. This plot right here shows by site on the x-axis the percentage of the reads that were generated by sequencing that were associated with different viral species. And so you can see this goes from 0 to, 90. And you can see for many sites, we had a very high percentage of those reads were viral reads. So 50%, or nearly or close to, in many of our sites. And even in these sites where we do have lower percentages of viral reads, they're much higher percentages than we would likely see if we were going to have if we were going to not enrich the samples. Sorry, my mind went blank. So we see here that the enrichment process worked very well in helping us predominantly get viral reads in these samples. And we detected a really wide range of viruses across all the different sites. The viral diversity that we recovered also compared well to previous studies. So this is WastewaterSCAN phase A. These are other studies from the published literature, previous studies using pure shotgun metagenomics where you just sequence everything, there's no enrichment. And then this is previous studies that are using that same twist panel that is enriching. Did I break up for a minute? I think I'm frozen.
No, you're good. You're good.
Okay, my screen isn't moving. So that's what's on the x-axis here. On the y-axis, you can see the number of high-quality viral detections that we have. And so we had a really, large number of high-quality viral detections in comparison to other studies using similar methods. And so we were really pleased to see that there is, a lot of viruses that we are detecting here. The results do have a lot of variance. That probably has a lot to do with the kind of effective specific sites. And also, we're comparing to the published literature where we're assuming that we're sort of representing a larger swath of our data here than what it kind of shows up in the published literature necessarily. So, anyway, really great viral diversity across our samples. So after classification using our pipeline, the follow-up steps depend on what pathogens are identified in the sample. So as we present in this preprint and as we continue to move forward with this work in phase B and perhaps beyond, we have divided the different viruses that we're looking at into a green list and a red list. So the green list pathogens are those that are more common. They're endemic to our study areas like SARS-CoV-2, RSV, norovirus, and also pathogens that are less common may cause some concern, but aren't really sort of shocking or surprising. They might be things that we're measuring by ddPCR, like measles or dengue. And the action for those is that they're summarized as part of the regular data output. So what you can see in this plot here is a summary of the green list pathogens that were detected for one of our samples. And I'll get into that some more in just a moment. The red list pathogens are those that are higher alert. They're classified as a select agent or similar. So things like Ebola, polio that, we understand everybody's very reasonably quite concerned about. The action for those is if our pipeline identifies that it thinks that one of those viruses is in there, we do a follow-up confirmation with direct inspection of the reads to make sure that when we compare closely, it really does align with that virus and not something that is similar or a viral vector or something like that. So for the green list, we're using that regular pipeline, taking that output, plotting it, and summarizing it. For the red list, we're holding that more closely, looking more closely, and then taking any follow-up actions that we would need to take, such as reporting, because of course, if we were to identify a select agent, then we would need to look closer into that and we would need to report it. So that is how we're splitting this out. And then what you can see in this plot here is for one of our sites, detection by sequencing in terms of a relative abundance measurement called RPKMF. So this is reads per kilobase of genome per million filtered reads. So that's, you can just say RPKMF after reading that whole thing one time, but this is essentially a measure of relative abundance. It's the number of reads for that are assigned to a virus, but also normalized by the length of the genome of that virus and the total reads generated by sequencing. So that means that we're robust to, if there are a lot of reads of a particular virus, but that genome is really long and there are the same number of reads for a virus that has a really short genome, it's normalized to account for those differences. And it's also normalized to account for if you have one sample where we have more reads that were generated than a sample the previous week that had fewer reads for some reason that were generated. It's normalized to control for some of those differences. So it's important to remember that this is a measure of relative abundance, that this is an enriched sample. And so there are a lot of things that can kind of alter the quantitative relationships and the quantitative amounts that we see over time. But through this heat map, you can see where we're detecting pathogens in higher abundance and lower abundance and how they track over time. For example, rotavirus A, this very dark blue, is highly abundant in pretty much all of the samples. And then you can look at, something like, para-influenza and see it's in lower concentrations here and not always detected. So this is a great way for us to view a sort of semi-quantitative output of the data for those green list pathogens. I see that there are a lot of questions that are getting answered in the chat. I'm going to let everybody continue to answer those and finish up with the presentation, and then we'll have more time for discussion. So finally, the last thing I want to show is this comparison of sequencing RPKMF and the measurements that we've made for some of these same viruses that we're measuring all the time through our normal ddPCR process with solids. Now, an important caveat to remember here is although these are the same samples, they're different parts of the same samples, right? We're measuring the liquids for the sequencing, or measuring the solids for the PCR. So it is not a direct comparison of the same material, but it's a relevant and important comparison for those of us who are looking both at ddPCR data and sequencing data from WastewaterSCAN sites, right? Overall, we saw that the relationship between RPKMF and the gene copies per gram ddPCR, as measured by Kendall's tau, was moderate to strong within each site. It was not as strong when we looked at comparing the same virus across a bunch of different sites. So looking at individual sites, seems like there was very much a site-specific effect. Some sites do have strong associations between the measurements, but low rates of detection by both methods. So Birmingham is an example here where we had fewer detections overall, and the viruses that are detected here are those that are typically in higher abundance. You'll see it's norovirus, SARS-CoV-2, and adenovirus F are really the main ones where we saw them in both types of testing for Birmingham. So we're also trying to continue to unpack, why might this process work better at some sites than others in terms of that viral read percentage I showed at the beginning, in terms of the detection of different species. That's definitely something that we still have more to learn about, but it was encouraging to see that the results that we get from sequencing tend to generally align with the results that we are getting from PCR. We love to see that. So we have this preprint that's available now. Please take a look at it. Feel free to ask us questions. We'll have time for some more questions after Amanda shares just a couple things with us. I know we have a lot of content that we're throwing at you today, so we're happy to spend more time on this, but wanted to give you an introduction to it now. And like I said, we are in phase B now, where we're working with 30 sites and starting to think more about how might we put this data to use for all of you, for public health. What does the future of sharing this information look like? So we'll be eager to hear more from all of you about that. And I will pass it to Amanda.
Yeah, thanks, Marlene. I'm going to keep this real brief because I want to make sure we have time for questions. So as a reminder, we have a categorization system. Next slide. We look at trends, levels, and frequency of detection. You can find all of this on our About page. I just put a link in the chat. Next slide. So as Ali mentioned, with the new pathogens, we recently onboarded TB and NDM. We are not using our categorization for that. We haven't assigned a category for the H1, H3 influenza marker or human adenovirus group F. So all of this is on our About page if you would like to see more information. So next slide. Okay, I'm going to just go over a couple of pathogens here. So, SARS-CoV-2, we typically see a summer wave start to happen late June, early July, and we haven't really seen that so far this year. We're slowly starting to see concentrations increase across some sites in the South, Northeast, and Bay Area, but levels are still very low. And in the last 21 days, we've only had a 65% detection rate for SARS-CoV-2. Next slide. EBD 68. So EBD 68 is typically a biennial nature to when we see outbreaks happen. And we saw little blips last year, but so far we haven't had many detections. So far in July, we've only had a 2% detection rate. So not sure whether we're going to start to see detections happen a little bit later into the summer and fall, but we will continue to keep an eye on this. Next slide. So I wanted to give an update on scanning the sewers. So we are going to have a session on antibiotic resistance genes in August. So we are going to have Amy Pickering from Berkeley and Amy Prudent from Virginia Tech. They are going to be giving two different presentations on ARGs in wastewater. We hope you can attend that session. I just included the link to RSVP in the chat. So please sign up and we will get you registered for the event. We currently do not have any programming for September through December. So if there is a topic you would like to hear from, I would love to know what that is because I want to make sure that these sessions are useful for you. So please send me an email. I have my email listed on the slide if you have an idea or if you have a topic you want to discuss. Next slide. Okay, lastly, in a couple of weeks is the California Department of Public Health West Coast Surveillance Conference, and that is going to be happening in Sacramento. So I think in-person registration is closed currently, but still virtual registration is open. So please feel free to register for this. Several members of the WastewaterSCAN team will be presenting. Marlene and Alex will both be presenting, and a number of us will be there too. So we'd love to see you in person or online if you are attending virtually. So I think that's it. I want to open it for questions. There's a lot of stuff in the chat right now. So
Maybe the